Now, boys, if you guys remember, in the last two classes, we have talked about the mouth and esophagus and stomach, right?
Yes, sir.
So today,
Yes.
inshallah, we are going to learn what happens in the small intestine. Okay. Now, boys, quickly, I'm going to draw the diagram.
Hmm.
So, boys, I just finished the drawing. Now, if you guys remember, in the last lecture, we reached up till the stomach and we learned that inside the stomach there is chyme. Do you remember this, boys? That this
Yes,
is
sir.
chyme. This chyme is actually it is the mixture of gastric juice and food. And I also taught you that as in the gastric juice there is pepsin enzyme which digests the proteins and break them down into peptides. Now, who will tell me? Who remembers what are peptides?
So peptides are 4 to 6 chains of amino acid.
Very good,
So,
excellent answer. Well done, son. 4 to 6 amino acid long chain, right? Understand? Now, was what happens next step? That this chyme then it enters into the small intestine. This is small intestine. Now, you have to remember that this chyme it is acidic. Why this chyme is acidic? Because it contains HCl. Now, if you guys remember, I also taught you guys that this HCl it is corrosive, right? It can burn the inner wall of the stomach. Now, if it can burn the inner wall of the stomach, it can still burn the inner wall of the small intestine as well as this chyme enters into the small intestine. So as it can burn the inner wall of the small intestine to protect it from burning, again Allah has provided the layer of mucus even on the inside of the small intestine. Understood, everyone? So even inside this small intestine, there is mucus lining. Now, if you guys remember, I taught you that in the stomach there is the mucus lining, and that mucus lining protects the stomach from the corrosive, nature of the, HCl. So boys, even this over here, this is the mucus lining. Now, this is one thing. Second thing, boys, that this organ over here, this is called as liver. Now, boys, liver is very, very important organs of our body. Now, how liver is very, very important? It performs many important functions in our body, like it detoxify the toxic substances. Now, question is, sir, what do you mean by detoxify? Means that it breaks down the poisonous molecules, poisonous substances present inside our body. Second thing, boys, is that this liver, it produces bile. Now, guys, let me tell you one thing, that the bile is a liquid that helps in digestion, but it does not have any enzymes in it. Bile has no enzymes in it. Now, first of all, I'm going to tell you about the composition of the bile. What are the things that make the bile? First thing wise, that there is 90% water in the bile. There is 90% water in the bile. Rest wise of the 10% contained the things which I'm going to tell you now. It contains bicarbonate and carbonate salts. Now, who will tell me where else we studied that there were bicarbonate and carbonate salts?
So inside the
Saliva. Right, boys? If you guys remember, we learned that these bicarbonate and carbonate salts are also present in the saliva. Next, boys, in the bile, there is broken down hemoglobin. Now, boys, if you remember that this hemoglobin, it is a protein which is red in color, which contains iron, and this protein is present in the red blood cells. Right, boys?
Yes,
Now,
sir.
what happens when this hemoglobin, it break down? We get two compounds. One, we get bilirubin, and second compound which we get, that is biliverdin. Now, guys, the first one, you have to remember the name of the bilirubin. If you forget biliverdin, no problem. But you have to remember the name of bilirubin, okay? Because this is the main component of the, which is of the hemoglobin broke down compounds. Right, boys? So remember that in the bile, there is 90% water. Rest of the 10%, it contains bicarbonate and carbonate salts. Then there is broken down hemoglobin, and the main part of that broken down hemoglobin is bilirubin. Another very important point that this bile, it has 8 pH. Now remember that the saliva also had the 8 pH. Saliva also had the 8 pH, and the bile also has the 8 pH. Boys, you have to memorize this. You have to remember this. Another thing which you need to remember, that the bile has bicarbonate and carbonate salts. Saliva also had carbonate and bicarbonate salts. So because of the presence of these salts, these have 8 pH. These have 8 pH. Understood, everyone? Did you
Yes,
guys
sir.
understand this?
Yes,
Now, guys,
sir.
what happens that this bile, it is made in the liver, and then it is stored in this pouch-like organ. Now, this pouch-like organ, it is called as gallbladder. Now, the job of the gallbladder is that it stores the bile. Now, when the chyme comes into the small intestine, right at the same time, the bile from the gallbladder comes into the small intestine, passing through this tube, which is called as bile duct. Now, when the bile comes in the small intestine, two things happen over here. Now, remember that the chyme is acidic. Right? And bile, it is alkaline. So when there is an acid and an alkali, what happens? Who will tell me?
They both
Sir,
neutralize.
neutralization. So
What?
neutralization.
Very good.
Neutralization.
Excellent. Neutralization reaction. So whilst because of the chyme, which is acidic, and bile, which is alkaline, there will be a neutralization reaction. There will be a neutralization reaction. Now, guys, which kind of neutralization reaction will be there? I'm going to show you, and I'm going to give you an idea. Now, you guys know that in the chyme, there is HCl. Right, guys? There is hydrochloric acid in the chyme. Then, guys, let's suppose that in the bile there is sodium bicarbonate salt. Have you seen the formula of sodium bicarbonate before?
Yes, sir.
Yes, sir.
Right? So there is this sodium bicarbonate. Now what will happen in this reaction? Look here, boys. This sodium, it will join with chlorine to form what? Sodium
Normal salt.
chloride. Got it? Then look here, there is this one hydrogen. This is the second hydrogen. So it will be what? H2. And then one of the oxygen will come to make what? Water.
H2O.
Understand? Now what is left behind? CO2. Right, boys? So what happened? The acidic chyme is now what? Neutralized. Acidic chyme is now what? Is neutralized. Understand, everyone?
Yes, sir.
So what happens? The neutralization reaction happens. Now, next step, what happens? Even after neutralization, excess bile comes, and that excess bile will make the chyme pH 8. That excess bile will make the chyme pH 8. So now what has happened was that in this stomach the pH was 2, and when the chyme came in the small intestine, after neutralization reaction, the pH became 7, but the bile kept on coming, and eventually because of this so much bile, what has happened? That the system, this you see chyme has become what now of 8 pH. Understand? Now, why the pH needs to be 8 over here? Now, boys, pH needs to be 8 because all the enzymes in the small intestine, they work at 8 pH. All the enzymes in the small intestine, they work at 8 pH. Right, boys?
Yes, sir.
Now, this was the first process. Now, there is this second process that happens over here, and that is called as emulsification. Now, remember that you ate chicken biryani, right? Now, the chicken biryani, it has carbohydrates, is right? Starch, we talked about it. Second, we talk about that there is proteins. We also talked about it. Third things are what? Lipids, the fats. Right, boys? Now, the fat digestion happens in the small intestine, but for that fat digestion, emulsification is required. Now, question is, what is emulsification? It means that converting large drops of fats into smaller droplets to increase their surface area. Now, boys, this happens in the presence of bile. Now, boys, what you can do, you can perform an experiment today at home. What you guys do, that you take, like you see, half a glass of water, put a lot of salt into it and mix it. Or what you can do, that you see, just first of all, you just take the uh, half the glass of water and put a drop of oil in it. But you will see that the drop of oil, it will be one drop of oil, big. After that, what you do, that you put some salt in it and mix the salt. What you will see, that large one big drop of the fat will change into small fat drops. So what is that happen? That happens emulsification. Now let me show you to give you a little idea. Let's suppose that this is a fat drop. After emulsification, this large fat drop, it changed into smaller fat droplets. Now, what is the benefit of changing this large fat drop into smaller fat droplets? And let's suppose that these red lines which are making, these are enzymes acting on the surface of fat drop. Now you guys can see that as there is a big fat drop, less number of enzymes are working. but as they become smaller in size, so on each small droplet, there are enzymes working to break it down. So what has happened? That the surface area has increased by changing the large drop of fat into small droplets. Large drop of fat into small droplets. So what has happened? That increase in surface area, and because of that, more enzymes are working on fat molecules, right? And the digestion of fats becomes faster. Did you guys understand this?
Yes, sir.
Yes,
Right?
sir.
So once two processes happen, one process is neutralization and second is emulsification. Now, as we come to this organ of the digestive system, which is called as pancreas. Now, why is this pancreas? It produces pancreatic juice. Why is this pancreatic juice? It contains three types of enzymes in it. It contains carbohydrases, it contains proteases, and it also contains lipases. Now, guys, very important thing that you ate chicken biryani. I'm reminding you again and again, right? Now, in the chicken biryani, there was starch. Starch is a carbohydrate, right? Now, if you guys remember that there was amylase enzyme in the saliva in your mouth, I taught you that. And that amylase also digests some of the starch in your mouth. Now, rest of the starch, it will be digested by the amylase enzyme. Now, one of the carbohydrates produced in the pancreas are again amylase. It means that amylase is not only present in your mouth, it is also present in your small intestine. So, starch will be broken down by amylase into maltose. Then there is the enzyme that breaks down the maltose, and the name of that enzyme is maltase enzyme. And it will break down the maltose into glucose. So, guys, what has happened? That all the starch is now what? Is digested, and we have got what? We have got glucose. Then, if you guys remember, that there were peptides or there were proteins. Peptide means, I just told you and reminded you today that the pepsin breaks the large molecules of proteins, large chains of proteins into peptides which are small. Now, these peptides or proteins, they are broken down by protease enzymes into amino acids. So whereas all the proteins, they will be broken down into amino acids.
Sir,
Yes.
is the name of this protease trypsin?
Yes, this is one of the trypsin is one of the enzymes. Trypsin is one of the proteases. Okay. Now,
Yes.
there are many, right? So trypsin is one of them. Then was there were lipids. Lipids are broken down by lipases into fatty acids and glycerol. Now, guys, if you guys remember, in the chapter number 4, we learned that lipids are made by joining of fatty acids and glycerol. So it is obvious when the lipids, they will break down, they will definitely make fatty acids and glycerol. Understand, everyone? Now,
Yes, sir.
what has happened that all the food which we ate is now broken down into what? Into more simpler food. We have got glucose, we have got amino acids, we have got fatty acids, and we have got glycerol. Now, guys, digestion is complete. Now, next step is absorption of the digested food. Now, guys, let me remind you something that if you guys remember, I taught you that the small intestine, we divide it into three parts for our understanding. We divide the small intestine into three parts for our understanding. Now, one of those parts is called as duodenum. Second part is called as jejunum, and third part is called as ileum.
Ileum.
So, whereas there is duodenum, there is jejunum, and then there is ileum.
Ileum.
Now, as let me tell you one thing: the duodenum has only one job, and that is digestion of food. Means that only food will be digested in the duodenum. Jejunum has two jobs. A little bit of digestion and little bit of absorption takes place over here. Digestion and absorption of the digested food. But in the ileum, there is only one job, and that is the absorption of the digested food. Right, guys? So in the ileum, there is only one process happens, and that process is the absorption of the digested food. Understood, everyone?
Yes, sir.
Yes, sir.
Now the absorption happens with the help of now as digestion, sorry, this absorption happens with the help of special structures which are called as villi. Now those villi, they are present in the ileum. Now let's suppose that this is a small part of the ileum. Now on the inside of the ileum, there are these finger-like projections. Boys, these finger-like projections, they are called as villi. This is the plural, and singular is called as villus. Singular is called as villus. Right, boys? Now, from here, inshallah, we'll continue in the next class. I'm just going to save this and share it with you guys in the OneNote. If you guys have any question till that time, you guys can ask me. Anyone has any question, you can ask.
No, sir.
No, sir.
Boys, you can find all the lectures in the, you say this content library, okay, in the biology folder. You see, I write down the date, and in under the date you can find. The lecture is shared, okay, boys? Okay, boys, see you guys inshallah in next class. Take care and Allah Hafiz.
Allah Hafiz, sir.
Allah Hafiz.
Allah Hafiz.
Allah Hafiz.
Allah Hafiz.